Hi, I'm Guillaume Thibault. I am a principal investigator at the School of Biological Sciences at Nanyang Technological University. Perhaps you're watching this video because you're considering to join our PhD program at SBS, and you might be interested to join my lab for your research. So I'm going to tell you in this video a bit more about how I got to, what was my path to get to SBS and have my own lab. And I will tell you a bit about what is the main focus of our lab, what we are mostly interested in, and the list of current PhD projects that are available. So I first started to actually study in chemistry quite a while ago in what we call a college in Quebec, which is a province in Canada. It is the equivalent to a polytechnic in Singapore. Then I decided to study further, and I got my bachelor in biochemistry also in Montreal, Canada. And then I moved to the University of Toronto, where I got my PhD in biochemistry in the lab of Walid Houri, where I was mostly interested by the molecular chaperone family HSP100, which is pretty much the equivalent of, it's the ancestor, if you like, of the proteasome. And then I moved to Singapore for my postdoc research in the lab of Davis Ng, where I worked mostly on ER stress response and the unfolded protein response upon lipid bilayer stress, or LBS. I'm going to tell you a bit more about what are these acronyms and terms, what they mean. And after a bit more than 5 years, I moved to NTU, where I started my own lab as an independent investigator. And I have been here since then. And I also have an adjoint appointment at IMCB A-Star in Singapore. So as I mentioned, we are interested by the endoplasmic reticulum. So just as a refresher, I'm sure you know that it plays a very important role in regulating calcium, because it stores calcium in the ER, and then it can be released to the cytosol. It's also the hub of lipid biosynthesis genes, including phospholipid, cholesterol, and sphingolipids. And it's also a very important place where protein biosynthesis occurs. It is actually 1/3 of the whole genomes that is translated into proteins that have to transit through the ER for the synthesis and then to be sent to the final destination. So you probably know that these proteins, 1/3 of these proteins actually, when they fold and they transit into the ER, then they will be transported to their final destination through the trafficking of proteins through vesicle transport. So you can imagine that if some of these proteins don't fold properly at the endoplasmic reticulum, it will have a domino effect on other organelles in the cell. So it's very, very important that this organelle stays functional and healthy, if you like. So why should we care about ER stress? It's actually associated with a large variety of diseases, and ER stress actually contribute to the progression of some of these diseases. So ER stress is actually classically is defined as the accumulation of unfolded protein at the ER. And when that occurs, it triggers a highly conserved stress response, which we call the unfolded protein response, or UPR. And when this response occurs, it attempts to restore ER homeostasis. And if that succeeds, the cell will survive. But if this process fails, it can lead to a cell death through apoptosis. There are actually 3 sensors of ER stress, which are highly conserved. So there is IR1, PERK, and ATF6. And overall, what they do is that they shut down translation for a temporary period, and they upregulate their own subset of genes from these 3 pathways. And those genes that are upregulated, it's an attempt to restore ER homeostasis. So those include molecular chaperone to assist in folding of unfolded protein. There's also lipid biosynthesis genes to restore lipid homeostasis at the ER. And in lower eukaryotes, actually only RE1 is found in lower eukaryote, including budding yeast and POMB. So I mentioned that ER stress is defined as the accumulation of unfolded protein, which is sensed by the luminal domain of RE1. So when it sends unfolded protein, it will activate the pathway where RE1 spliced HAC1 messenger RNA, which is then translated into an active transcription factor, leading to the upregulation of about 400 genes in yeast. More recently, other labs and also my lab as well, we have contributed to the fact that there's an alternative sensing of ER stress through lipid barrier stress, or LBS, which is independent from the accumulation of unfolded protein, where RE1 can specifically sense lipid barrier stress through changes at the lipid barrier that is sensed by the transmembrane domain of RE1, and then the downstreams is similar to the activation from unfolded protein. So in my lab, we are mostly interested by this phenomena of ER stress and the unfolded protein response that is activated from lipid barrier stress. And this is actually very, very important, especially in the context of metabolic diseases, where we have a good sense that this is a big contributors of ER stress in those conditions, but it's still poorly understood what is actually the definition in those contexts of diseases of lipid barrier stress, and how RE1, what does it actually sense to activate this pathway. So we have different projects currently open in the lab, including mostly, here I wrote cellular stress response, but it's mostly ER stress response. And there are other stress responses that in the cell that occurs, including heat shock response and mitochondrial stress, that we also interested to look together in the context of the unfolded protein response. So I invite you to scan the QR code to have a bit more details on the projects. We're also interested to study the role of ER stress, especially in the context of aging, and looking at how RE1 sense ER stress, and more precisely lipid barrier stress during aging. We also have another interesting collaboration where we looked at the role of ER stress in the context of wound healing, and more specifically during infection. And finally, we have an ongoing project where we collaborate with a group in China to dissect the minimal pathway that is required to restore ER homeostasis in mammalian cells, which is very difficult to do in mammalian cells. So that's why we are using yeast, where we bring the pathway from human to yeast to identify and isolate the minimal pathway that is actually required to restore homeostasis. So if you have more questions, you want to know more about our lab, I invite you to our website. You can also scan the QR code or just type the lab address here. You can also contact me. You will find my email address from the website. I'm also open to other projects if you have some ideas, so that's something we can always discuss. Please, just contact me, and then we can have a discussion from there. So here is a picture from July 2019. I don't have a picture from this year because of COVID, but here are the current lab members, and we have some collaborations going on, and you can find us on different social media. And this is our current sources of funding to support the research in my lab. So thanks for watching, and I look forward to hear from you. Thank you.